Good morning, madam. Started, madam. Good morning, Padmavati, madam. Live started.
Okay, Basha. Thank you. Good morning, everyone. A warm welcome to fourth day session of Faculty Development Program. I'm so pleased to be with you this morning and have a chance to introduce the speaker of today's session, eminent scientist, Dr. Kantikiran Varanasi, Head Preclinical and Bioassay in Global Clinical Management, IPDO, Dr. Reddy's Laboratories Limited, Hyderabad. In his current role, Dr. Kantikiran is responsible for designing and management of various biostudies across clinical and non-clinical and clinical development of various differentiated and complex generics across global market. Dr. Kantikiran holds bachelor degree in pharmacy and master's with pharmacology specialization from Andhra University. He did his PhD at Birla Institute of Technology and Science, Pilani. As a management aspirant, he holds additional masters in business management from Indira Gandhi National Open University, New Delhi. He has over 22 years of research experience, worked in the areas of biopharmaceutics, DMPKPD, toxicokinetics, bioassay, bioanalytics, microbiology, CPPK for both discovery and formulation research in areas of oncology, metabolic disorders, pain, and inflammation pathways. His career started with Dr. Reddy's Research Foundation, Hyderabad, in India, in 1997 as a pharmacologist, where he was worked in the areas of diabetes, hyperlipidemia, cancer, anti-inflammatory research to screen and develop the new chemical entities using various pharmacological assays. In 2002, he joined Sun Pharma Advanced Research Center, Vadodara, India, as a senior executive pharmacokinetics and clinical pharmacology department, where he played major role in development of clinical pharmacology unit and established international regulatory development. In 2003, he moved to Glenmark Pharmaceuticals Limited, Navi Mumbai, worked as a principal scientist and head pharmacokinetics and drug metabolism, where he established several in vitro and in vivo ADME techniques. In 2002, he moved to Incozen Therapeutics and served as a director, ADME and safety metabolism. In 2012, again, he joined Dr. Reddy Laboratories as an associate director. In addition, Dr. Kantikiran was also trained in various GCP and GLP workshops and an internal quality auditor certification program. Dr. Kantikiran has published various peer-reviewed articles in peer-reviewed international journals and have several abstracts, patents, guideship to his credit. With this, I request Dr. Kantikiran to start his riveting session and hope this session would be enlightening and informative to everyone. So now I request Dr. Kantikiran sir to proceed with his session. Thank you.
Thank you, madam. Thanks for your nice introduction and good morning to all. Dear faculty members and students and some of the industry colleagues who are participating in this seminar. I would like to thank you and warm welcome you for this session on a brief presentation on the rapid bioanalysis in drug discovery and development, which I am going to talk today. So today we are going to discuss about the slight overview of drug discovery process and what is the role of bioanalysis in drug discovery and development, how these bioanalytical methods are to be developed, and what are the validation parameters need to be considered once a method is developed, and what is the flow of the sample analysis and reporting in the subsequent step to the method validation, and what are the regulatory guidance or regulatory science driving this area of research. I know most of you are working either fully or partly or in the areas of like drug discovery and development. Some of you might be academicians, some of you might be students aspiring to work in this drug discovery and development, and some of you might be already working in the research and development and operations of this whole drug discovery and development. Now we know the importance of our area, and which is in the current situation of the the COVID also, how this industry is playing a major role, and at the same time, how industry is in fact trying to get that gained information and then putting data extracted, and then how quickly, if anything, need to be performed in subsequent to in addition to what we need to bring to the market. So how rapidly we can operate and then get into the market in the need of the time. So that's where so uh, one of like when we are talking about the bioanalysis in this particular research area, how rapidly we can develop and validate and then come out to the completion of the studies in time in a way that it can enable the products to get approved by the agency and subsequently can reach to the market in the critical need of the time. So that is a very much importance of the rapid role of these areas. Hence, I believe this is very much applicable in the current scenario. So let us go back to the overall view of this drug discovery process. Most of you, I know every one of you might be aware that what are the different stages of discovery and design. And just to start with that, yes, it starts with for any new chemical entity or new biological entity or any new product or new for dosage formulation. If we are developing, we need to look at that various aspects. So it's when it starts with a new chemical entity or new biological entity, so the medicinal chemistry, which is playing a role, or the bioinformatics, which plays a role in terms of the NV. So it starts with the discovery operations, where typically we can refer to the NCE area for our ease of understanding, and then medicinal chemistry, where it designs the various pharmacophores, and they will identify the pharmacophores and design the structure and make them synthesizable. So once it is being synthesized to the required quantities to be tested subsequently in the battery of the test. So once it is being like a idea generation and then subsequent synthesis, and whatever the synthesized molecules will next go into the biological characterization, where it will be subjected to its target uh, whether it reaches the target potency or not. So by in vitro biological techniques, and subsequently any preliminary pharmacology studies can be understood very quickly. While doing so, it is imperative that the drug metabolism and pharmacokinetics, which is nothing but the when you study of this absorption, distribution, metabolism, and excretion of the drugs, and at the same time safety. So that's why we call as ADMET properties. Before once the compound is synthesized based on the structure identification, and then based on the whatever the structures available, once it you can apply the principles of ADMET, which will enable the molecule to be available in the in vivo system, such that it can perform with the adequate efficacy and within the principles of safety based on this ADMET property. So the one step is the lead identification, what we discussed from chemistry to biology, and then once out of the several molecule, once the lead is identified, and further it goes to a development area where we need to look at that what are the leads, and those leads can be developed very clearly, whether a battery of tests which need to understand extensively with that this primarily for safety and subsequently for the efficacy. And so preclinical development need to be performed with this lead candidate, and subsequently once it is being proven and well efficacious and pharmaceutically enabled compound, then it can it can go into the subsequent translation research into the by moving into the clinical development. So translation research here, it connects between the preclinical to clinical by understanding various aspects of what are the things it will translate from the preclinical models to the clinical studies. So that's where the core area of translation research, and subsequently the molecule can enter into the clinical phase, where it can go into the standard battery of clinical phase 1, 2, and 3, and post to 3 again it needs to be applied to the agency, and then subsequent new drug approvals. Once it is approved, and then it can go into the market, and again post market studies like phase 4 studies will be enabled. So this is the overall process. Everyone is aware of that. It is very important to understand what are the resources, time, and everything we need to understand about that. So if you look at that, so how we can move into the clinical development. Once if it goes to the clinical development, not all the molecules will be successful in the clinical development. So it takes, and at the same time, the clinical development pathway is a regulatory pathway, where it goes irrespective of the designs and the planning what you do, but based on the agency's reviews and subsequent to that, there is a standard time which is applicable for this either preclinical development or clinical development. So the most important part aspect for a scientist is to research scientist is to look at that whether I can reduce the time in the early discovery process itself, so that we can proceed with whatever the standard norms of the timeline for the development time, and subsequently it makes it successful. So again, as we discussed already, that if several molecules are being taken the medchem, and subsequently they will go to in vitro biology or in vitro characterization, and at the same time parallelly biopharmaceutical properties where stability, permeability, lipophilicity, protein binding, solubility of the drug, all these things need to be understood as we discussed that the like in the principles of ADMET, in a way that it will make the compound to be available in the in vivo when it is being administered. So quickly, if we can try to understand these biopharmaceutical properties and go to the DMPK, where again in vivo studies where one needs to look at that what is a single dose, what is the multiple dose, how about the species effect. While understanding that, there are the other aspects like metabolic stability, CYP inhibition, which will enable the molecule to be successfully um, uh, 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 get developed without much interactions or much what we can say is a drug-drug interactions, to avoid the drug-drug interaction in the which might or which may limit the development of this candidate in the future scenario. So once these are being understood, a proper preclinical tox studies, safety studies need to be understood, where the validations of all the methods established prior to that need to be generated and subsequently analyzed for the subsequent development. The most critical part here is the time. Okay, so the timely support is very essential to improve the success rate of these molecules when they are moving along the path of from the early discovery to the clinical development. So one needs to understand that how we can tailor out, and how one at the same time how quickly and efficiently we can move to different stages. So this is where area one need to focus on that, and at the same time in subsequent slides we'll talk about how bioanalysis will play a role in this time factor. So what is the bioanalysis? As we know that bioanalysis is nothing but a subdiscipline of analytical chemistry, covering the quantitative measurements of xenobiotics, which are maybe drugs or their metabolites or any biological molecules in unnatural locations or concentrations. At the same time, the biotics, which are nothing but the macromolecules, proteins, DNA, large molecules, and metabolites in biological system. So overall, this is a science which is able to characterize the exposure of the drug or the biological entity in a given biological matrix in such a way that it will be helpful to understand further its pharmacokinetic properties. Okay, so where this applies, the application is majorly in all the areas of drug discovery and design, drug development, and formulation research. We already talked about discovery, design, development. So how it is developed. So once again, another core area of interest is formulation research, where most of the Indian pharma follows this path, either by making a generics of the existing marketed products or differentiated products compared to the existing marketed products. So in both ways, one needs to establish, rather than going for with a new formulation, one need not to go with the entire process of the development again. Rather, he can generate the bioequivalence or bioavailability study, comparative bioavailability study, in such a way to substantiate that the exposure ratios are same, in such a way that it will behave exactly as similar to that of the marketed product. So again, in this area, this bioanalysis plays very major role, because when you talk about the bioequivalence, so one needs to understand that the pharmacokinetic properties, whether it's like a systemic exposure, AUC, or it's a Cmax or Tmax, of everything need to be matched within the 80 to 125 criteria, where it is very, very important to understand the methodology what you are following to estimate these plasma concentrations or in any given matrix. So this bioanalysis plays a very critical role in establishing a very precise, adequate, and rugged method in a such a way that your exposures, whatever you estimate out of the studies, should be a true representative of the sample, and such that the decision can be arrived at that whether it is equivalent or not equivalent, how it is compared to the existing formulation. This is again same pathway what we have discussed, like right from the discovery to preclinical and phase one. In the phase two, again phase one, where you will be taking up the healthy subjects and trying to understand the pharmacokinetics and safety of the new molecule, and in subsequent to that phase 2b, where it will be in the subject population, where in the patients very a subset like a phase 2a studies or phase 2b studies depending on the size and indications. And at the same time, when it goes to phase 3, it's a multiple geographical like locations. Even the pharmacogenomics will play into in this area. So all these things leads to further approval and launch. So the bioanalysis plays right from the discovery to almost, I can say that, up to phase 2 completely. Of course, phase 3, when it goes to the pharmacogenomics and other things, obviously it plays a role in phase 3 as well. And of course, once it is launched, again, it is as good as applicable in the post-market studies as well as the phase 4 study, where we talk about for the generic or again for the differential product. This plays a very critical role. So the main goal of the bioanalysis is to support pharmacokinetic or toxicokinetics parameters in determining the ultimate product safety profile as the primary focus, and at the same time to understand its efficacy as well. So the core focus would be around developing the methods for lead compounds identified during the discovery stage. And next focus of the bioanalyst would be the validation of these methods, whatever is being developed, and they should be formalized and mandated as per the required norms during the preclinical development. And subsequently, again, bioanalysts should be able to enable, like transfer these methods or develop the methods for the human samples, which further need to be supported in the phase of the clinical development. So again, one may look at that, what are the species differences and other things, and then subsequently look at that these assays can be developed with very robust methods, and subsequently it can withstand the whole clinical development time. Of course, there are the couple of the changes might happen depending on the nature of sample, depending on the nature of the unknown peaks, other things. So the methods should be performed in a way that we can tailor out the method in a way that it can be slightly optimized in order to again troubleshoot the method in a way that we can get our objective to the suitable requirements. So for all these things, one need to develop the method very efficiently, and the method should be able to almost, when you are developing a method, it should be in such a way that when you validate the method, it can be validated easily, rather going back to the method. Like once it is failed, if the validation fails, again we will go back to the method development, look for the alternative methods, or again changing the methods. Rather than that, it would be better to develop a method very precisely, accurately, and to the extent possible rugged. Okay, so what are the criteria need to be understood while developing a method in the bioanalysis? So of course, what we are going to analyze is our analytes of interest, and the analyte could be a drug or a biological entity, or it can be any biomarker. So one or are we analyzing both the parent drug and metabolite? Are there a single analyte? Are there a multiple analytes we need to monitor depending on your, for example, if you are doing a combination studies where you need to analyze multiple analytes. So depending on the requirement of the method, one needs to look at that, what are my analytes of interest and what are the properties. So we need to look at that, what are the physicochemical properties of the analyte that could impact on your method development. So that need to be understood. And what are the samples we are going to analyze? For example, are we going to do a study in plasma? For example, are we going to collect the plasma and analyze the sample? Or are we going to study the pharmacokinetics in urine? Are we going to do the estimation of the drug in the fecal matrices, or any other matter, or any tissues, whatever in tissue distribution study, you might end up with the vital organs to all the organs of interest based on the pharmacodynamic and safety profile. So one needs to look at that, what is my matrix, and how best I can use that analyze the drug by cleaning the sample and subsequently analyze very selectively about the analyte. So so we need to look at that, what sample we need to, how the sample need to be characterized, how it need to be understood, and at the same time, since we will be estimating the drug, which is maybe a known or unknown drug with a different concentrations, because once if you administer any drug into the biological system, it follows its time course of action. So in a plasma, you will get a different absorption, distribution, elimination in such a way that you will get a different levels of the drug over a period of time. So when we are estimating an unknown drug over a period with a different levels of the drug, how best I can make my sample or our sample in such a way that we are analyzing the sample accurately. So we need to have a other standard, which is internal standard, which can be spiked at the same concentration in a way that to talk about your sample nature and ruggedness of the sample when you are analyzing them. So what could be the best internal standard, and whether it is ideally you need to select a standard based on your the structurally similar compound or same category of the compound, or the best one is by looking at the deuterated compounds of the same analytes, so that the sample could behave very similarly to that of the analyte of interest in a given system when whichever you are developing that. So if both the analyte of interest as well as internal standard behave exactly similar, then it will be very much easy for you to characterize your sample peak characteristics and other thing, and subsequently your whatever the concentration over a period of time what you estimate out of this activity could be more precise. And then subsequently, what are the so for analyzing what type of the chromatographic methods we need to choose, and then what are the instrument we need to select, and selection of the mobile phase, how it impacts depending on the whether we are using HPLC system, you are whether you use a mass detectors. So it at the same time what kind of the chromatography we are using. So it all depends on based on your methodology, what your sample separation techniques are cleaning up, you are choosing that. The mobile phases will be choosing that. At the same time, in order to selectively analyze your analyte of interest, what columns need to be used, how the column performs, and subsequently how best we can get out of our selective area of interest. At the same time, choice of detector, as we already discussed about that during the instruments, what kind of the detector it could be, like whether it's a UV or a spectrophotometer, or it can be a mass detector. So depending on your requirement of the analyte and the concentrations, what you could achieve through the biological samples, so that your choice of detector you need to select. At the same time, limit of detection, once if we are developing any method, are we getting what are my detectable limits, minimal detectable limit, in such a way that it could be free from the blank response, because every plasma will have its own chunk of interfering components which could play a role in getting absorbed, means to show your lambda max at certain time, or so whatever the blank response we will be getting out of that, to that whether my analyte of interest could be differentiated very well in such a way that what we call generally is the signal to noise ratio here. What is the signal of the, what is the noise of this background effect from the blank plasma? What is the signal of the analyte of interest? Then is that signal to noise ratio is 3 times, 5 times, 10 times, depending on the range what we look at, we will define the limit of detection and subsequently limit of quantifications and other aspects parallelly. So once the limit of quantification and subsequent and till what level from lower concentration to and higher concentration, till what concentration, upper concentration, it could follow a dynamic range. At the same time, it follows a linearity and then follows the Beer-Lambert's in a way that beyond a certain higher concentration, it could lead to saturation in such a way that we can avoid we can define the range of this linearity curve, such that your unknown concentration can be predicted very accurately. So this dynamic range is very important. It depends on your method, what you choose, whether you are using HPLC UV or whether you are using mass detector. So suppose, for example, if you are using a mass detector, Ideally, your dynamic range should not be more than 1,000, whereas in HPLC, one can go with a linearity range from 10 to 10, 10 p, for example, 10 nanogram per ml to, they can go up to like a 10,000 nanogram per ml or beyond that. However, in mass, to the extent possible, we need to look at that not more than 1,000 dynamic range, what we need to look at that. So these are the certain critical aspects we need to be considered based on the instrument to instrument and the your methodology, and at the same time how the sample behaves in that particular methodology. So the main interest one need to develop is, first of all, rather going blindly with that available techniques and try with that. At the same time, one needs to look at that. What is the molecular properties of a lead? Whether it's what structure it is, how it is like behaving. Is there any literature evidence for this particular structure? In any whether we can, for any bioanalytical scientist, before going into that, he can also look at that. What are the general analytical techniques when it is being analyzed in the aqueous sample as well? So those literature can be taken as a reference. At the same time, the molecular weights, its solubility, polarity, pKa. So all these things, once if you know that in advance, then you can define or design your the separation techniques very, very efficiently. So the polarity plays major role, because most of these drugs generally this drug compound may be polar or non-polar, but in most of the cases, most of the drugs in current market are under development are typically based on non-polar in nature. Hence, we need to understand the polarity aspect, and so that such that the method, whatever you can develop, could be tailored to the need of these based on its polarity. So, in general, anywhere if the molecule has a propensity of showing that less than 1.5 log P, then it is considered to be very polar, and 1.5 to 4 it is moderately polar, and whichever the compound shows the log P greater than 4, it is generally considered to be non-polar. So when we are looking at that polar to non-polar, then we need to look at that what are the solvents, and whether the solvents is that based on the miscibility of the this compound. So we can choose that water miscible solvents or immiscible solvents. So here is just example for you to understand that what are the miscible solvents like water, methanol, IPA, acetone, ethyl acetone, and what are the non-polar solvents. For example, like ethyl acetate is either THF, dichloroform, taurine, isooctane, hexane. Several others are there. These are the commonly used immiscible solvents, which are nothing but which are having the propensity to from the which are of the non-polar in nature. So once if we understand the basic physicochemical properties of this drug, the next step is to techniques need to be followed for a developing method. In general, the bioanalytical techniques are largely categorized, broadly categorized into three techniques. Sample preparation techniques, which are nothing but how do you clean up your sample in such a way that you only your selectively you can analyze your analyte of interest. And sample separation techniques, again, just in spite of the cleaning, still the interferences and other components might interfere. So how will you separate from the parent of interest from the interfering peaks? So it involves the separation chemistry, where you'll be using that what are the kinds of chromatography, and then what are the columns, and subsequently once it is adequately separated out, then how do you detect it? So these are the and we need to understand that what are the detection techniques, what detection technique is suitable for this particular drug, at the same time for this particular drug at particular concentration of requirement, that need to be understood. So we will briefly touch upon the various preparation techniques and separation techniques so that we can move forward. When it comes to separation techniques, which are nothing but the cleaning techniques, of course, there are the gold standard methods widely used in the industry are three primary techniques, which are nothing but the protein precipitations, liquid extractions, solid phase extraction. So now, what is the sample here? Sample is nothing but the matrix what you are choosing, whether it's a plasma, urine, serum, blood, whole blood, or fecal matter, or any particular tissue, or any other cell, even if you are trying to understand from the cellular components as well, or if you are trying to understand from the in vitro cell-based assay as well. So this so it depends on the matrix. All these matrices do contain again majorly they contain proteins and phospholipids and other lot of other components which might interfere with your analyte of interest when it is being either spike into that, or when it is being already when it's being administered to the in vivo system, and then already it is being absorbed and present in the specific matrix. So how to clean up this sample? So in general, if it is a polar drug, so and at the same time, if the analyte might be at the very high concentration, the simplest technique people follow is protein precipitation, because you can choose any water miscible solvents like estronitrile, methanol, and other solvents which we have discussed in the previous slide. So those things can be added to this sample in such a way that so these solvents, these are nothing but the organic solvents, but these are water miscible organic solvents. When it is being when the plasma or the matrix, whatever you choose, serum or blood, when it is subjected to these organic solvents, obviously whatever the proteins and other components will be denatured immediately with the high concentration of these organic solvents. So once it is denatured, so what it forms is the precipitation forms because of the denaturation of protein, and that precipitate can be well separated out by either by sedimentation or by centrifugation process, such that your the polar drug, whatever is there in your sample, could be easily come into the miscible portion of the free fraction, and that free fraction can be separated out and be analyzed. So in similar manner, these protein preparations are actually very fast in nature, so they are less laborious, and then but selectivity could be slightly bad, we can say that, compared to other technique because of the polarity and non-polarity issues. And at the same time, sensitivity can be good depending on the concentration of what you can try to achieve depending on the nature of the compound. But at the same time, this ion suppression is one of the factors where it could play a major role. It will use high ion suppression, and these are generally manual process. At the same time, like automation can be possible, but the low throughput will be applicable in this case. And majorly, the suitability for this is for the mostly like hydrophilic compound. Cost is of course low. So whereas when it comes to the other a better technique other than protein precipitation is liquid extraction. It mostly applicable for the lipophilic compounds, where you will be adding the water immiscible solvents, so which are trying to extract this non-polar drug of choice, and then subsequently, like for example, like ethyl acetate, dichloromethane, or TBME, these solvents, once if you add that, they will try to based on the partition base, they will try to extract the drug of your choice into so in your free fraction such a way that first of all you can separate it, you can very well extract it. So the first of all, this extracting solvent, they will pull out the drug from the sample matrix into the because based on the non-polar in nature, they will extract the drug into that particular solvent, such that when it is separated out, and so the sediment whatever is there can be discarded, and whatever the extracted solvent system containing of this drug can be evaporated, such that because these immiscible solvents are, since they are organic in nature, mostly they can be easily at particular boiling temperature and other things, so they can go into the evaporation such a way that the matrix containing your analyte of interest will be formed as a deposit at the bottom of the tube, and that can be again resolubilized or reconstituted with a ideal suitable solvent or suitable mobile phase which you are going to inject onto your chromatographic system. So that can be used, and then either directly or again with the modified techniques can be understood. So overall, this LLE is a very a good rugged practice and widely used in the industry. Before coming into this solid phase extraction, the other technique, see, this LLE plays a role very significantly over the past decades. However, yes, the current group of scientists or a current era is majorly focusing on solid phase extraction because of some other advantages. So otherwise, this LLE is more, yes, of course, it's more laborious because one needs to, if you want to extract it, you need to add the solvent and you need to put it for either using manual extraction or automatic liquid extractors. So it follows a certain labor process, and but at the same time, it is good for selectivity and sensitivity, and the ion suppression is low in this case compared to the protein precipitation, and automation is again it is again possible. In general, it's considered to be low, but it is possible, right? As we discussed, that is mostly suitable for the hydrophilic compound, and cost is high. And one disadvantage with this LNG is because when you are trying to extract into a organic solvent which are non-polar in nature, so the environmental factors could play in a way that, what I mean to say is, the environmental contamination with these sample preparation techniques will lead to a exposure to the human being, whoever is doing this analysis or any other, and at the same time it can impact on your some of the detectors and other aspects. So of course there are the ways to handle the samples in using fume hoods and other thing, but in general it is considered to be like not like a compatible for the greenery of the environment. Okay, whereas if we come to the other extraction technique, which is very widely used and current practice, I can say that most of the 80% of the current pharmaceutical industry depends on this extraction is nothing but solid phase extraction. Here you are having a solid phase extraction in a way that like it consists of cartridges which are having a solvents either from 50 to 200 in nature, and which will try to retain the choice when the drug is spiked in the matrix is being spiked into this one, the sorbent, and it can, depending on its nature of the molecule, it will go and it will retain on the sorbent such a way that all the other when you wash it properly with the other solvent which can mostly solubilize the other interfering substances, so that can be washed off, so that selectively your drug of interest as well as the maybe similar kind of compounds will be retained over this bed, and again it can be again washed back in a way that it can be purified to the extent possible, and then it can be eluted again with a suitable solvent for that particular drug of choice. So majorly if you look at this diagram where the SPE here, the major steps in the solid phase extraction, you need to wash this sorbents in advance so that you can precondition the column, and subsequently you load your drug of your sample and whatever is retained, and then wash that whatever the interfering peaks when it is being when the drug of choice is, once it is conditionized and it is absorbed onto the surface, onto the your sorbent surface, then subsequently you can elute that. So these are the washing, loading, and again washing and elution. So this is in a way to, one is the cleanup sample very efficiently, at the same time you are enriching the concentration of the your analyte of interest such a way that, since because you are washing multiple times and then you are doing that, so you are cleaning in a way that in a given matrix you are enriching the sample. So that's how this solid phase extraction helps in making clean and concentrated sample, which can be easily detected and which can be predicted well for the pharmacokinetic properties. So again, its cost is considered to be high at one stage earlier. Now it is considered to be, in fact, this is in current it is considered to be moderately high. I can say that, not high, low to moderate high. At the same, because a lot of manufacturers have come anywhere between any column, like it can range from 100 to couple of the hundreds. However, with almost now the competition has emerged in a way that the column cost is also coming up with a more bulk volume and other aspects, the way you negotiate with the vendor. So overall the cost is considered to be now moderately high, and at the same time it is highly suitable for both the hydrophilic and as well as lipophilic molecules. And the major advantage is it suppress the ion suppression effect. The impact is very low, and such that it can be easily suitable and adaptable for especially for the mass detector, and the sensitivity is very good and selectivity is very, very good. So leaving out these the standard gold standard techniques, there are the other techniques which are in market and are being used by the industry people, depending on the nature of the sample, complexity of the your product. So there are multiple techniques, which we normally consider them as a novel approaches, but at one stage these are novel approaches, but now I can say that these are not very novel, but at the same time these are less practiced depending on the nature of your product or the method or complexity of the method. So there are the advances in terms of the solid phase, which led to the micro extraction techniques. And so that, for example, you take whether it's a supported liquid extractions, like a LG with a SPE, or at the same time, there are the plates which are available to directly remove the phospholipids so that you can clean up the sample, and then you can either subject those samples to further liquid extraction or subsequently to the SPE, such a way that that sample can be easily um, uh, concentrated, and then selectively it can be analyzed. And there are the techniques using the magnetic beads, turbo flow, monolithic spin column extractions, and micro extraction, as we discussed about whether it's a packed sorbent or other techniques. So the micro extraction technique is getting widespread nowadays, based on the more availability of the vendors. At the same time, more awareness among the scientists to suitably take it for the subsequent sample nature, especially maybe, for example, if you are looking at that any kind of the liposomal formulations, a platform technology like microsphere samples, means dosage form containing microspheres or liposomes, or any other nanoparticles like nanomedicines, which you are making the special delivery system. If those analytes of interest, if they are not in a very free form, they are in the the texture form, in a way that what you call is like a bounded form or the free fraction. Those things can be looked at that depending on the need of your sample. One can look at that what type of the this separation technique I can take, preparation or separation techniques we can take further, and then we can use this judiciously, the new techniques, whether it can differentiate compared to the gold standard techniques. Of course, one need to look at that. One is that SP is good enough. If not, what kind of the additional advantage, like advanced techniques, can be taken further. And last but not the least is the dried blood spots. It has been introduced almost a decade ago. However, yes, it is being used by some of the industry counterparts, but not widely used, because still industry goes with that separation technique like a plasma or serum, and then analyze the samples. However, otherwise, these bright blood spots, there are the couple of the plates, are ready-made plates are available, where you can just put the spot of your blood in a way that the, these coated plates, they are inherently having this separation technique, and then that whatever the portion, in that portion you can take it out and you can extract it properly, and then subsequently you can analyze that. So these are all the new techniques. Depending on the usage, one can select it and judiciously use these new techniques. Otherwise, gold standard techniques can be used. So at the same time, one needs to understand that these are various techniques, but how do we rapidly develop a method? So we need to look at that. Yes, can I go sequentially, whether like a PPT, like a protein preparation, followed by LNE, followed by SP? No. So, as we discussed earlier, that the time is very important, and you need to reduce the early discovery process such a way that one should be able to develop a method within a day to the extent possible, because it actually it is you can understand the hardship, or at the same time challenges of the medicinal chemist when they synthesize the compound, because trying to convert the design into what you call is a powder form, which is a physical form, is very difficult when it is being getting synthesized. So when they take up certain time and then come out with that, so if we can reduce our analytical time, so that we can quickly give the feedback to the medicinal chemist about the pharmacokinetics nature of that particular molecule. So one needs to look at that, how best we can reduce the timeline of this method development. So then they can consider that whether we can take parallel approach of maybe subjecting the samples immediately once it comes. Maybe you can use parallelly for protein precipitation, liquid extraction, or SP. One can design parallelly in efficient way, in a way to reduce the time. At the same time, one also need to look at that what are the separation techniques. Again, how adequately and rapidly we can follow the separation techniques to reduce the time. So in general, the separation techniques are considered to be like either chromatographic techniques or chromatographic properties, where one needs to look at the various aspects. So majorly, the industry, as we know, the liquid chromatography falls into majorly for the reverse phase chromatography or normal phase chromatography, depending on the polarity of the compound. So for the reverse phase, it's mostly used for the non-polar compounds, where a non-polar stationary bed will be there in such a way that your non-polar compound will retain onto the non-polar bed, such as other polar interfering piece will get washed off when the either mobile phase or whatever is being flowed through that particular column, in a way that you can selectively retain your analyte of interest, and subsequently it can be detected. So in bioanalytical industry, now it is very well understood the analytical chemistry, generally they follow the normal phase, whereas the bioanalytical chemistry, uh, majority of the most of the scientists follow the reverse phase, because most of the drugs are non-polar in nature. So, but when the reverse phase is not working, one needs to look at that what is the other options like normal phase and other things, and then subsequently select the separation techniques and their mobile phase accordingly. At the same time, the columns which are used for this separation need to be considered based on your the technique what you follow. So columns, again, as we know, the widely used columns are this reverse phase, since we follow the reverse phase chromatography. So here C18 columns, C8 columns, and C4, C2, and phenyl bonded. These are all the widely used reverse phase columns. At the same time, there are the, when it comes to normal phase, silica, amino, and alumina based columns will be used. Of course, there are multiple columns, and one can look at that. It's entirely column chemistry is a very big area of research. So I'm limiting myself here on the types, and subsequently how best we can efficiently use those things, and we can look at that our detection systems. So once it is being detected on a detector, the response what you get is nothing but, here we call it as a peak. So how that peak looks like. So because what is our measurement? Here our measurement is peak response, either peak height or peak area. These are the two things normally to be considered for the peak response. So when we are taking the peak height or peak area, the peak should be very sharp and without any other aspects, like we can discuss about that. Normal peaks, the peak should be sharp and precise in a way that either height or area can be considered. And most of the cases, in general, we consider for the area rather than the height in general practice. It's not essential to go with that area only, but it's a general practice. But so when it comes to area, when you are analyzing that, depending on the peak shape, the area of the curve is nothing but the peak area of the curve will behave. So if the peak is very sharp, so your area will be very precise. If your peak is broad, then your area will be more, in a way that probably just imagine a situation where two analytes, one of your analyte of interest, another which is a unknown peak or your unwanted peak, is just eluting at the same time on the column. So both of them will merge together, and in your detector it may show a broadened peak. So it will bias your prediction in a way that we will be estimating the peak area based on this one. So hence it is very important to understand whether the peak shape is sharp, is it broad or sharp, or it is having any split at the tip. When it is being split, that means there is a second peak which is interfering in such a way that, yes, it is not as equal to that of the primary peak, but at the same time it is less, but it's merging. So the split of the peak need to be considered. At the same time, there are the whether peak fronting is there, peak tailoring is there. So fronting in the sense like again interference of the peak just before the peak of interest in a way that it broadens the front side. At the same time, peak tailoring in a way that it some of the portion or of the next eluting peak, it might be just coincidentally or is co-eluting in a such a way that it is broadening your the peak. So at the bottom. So in a way that the tailoring, what you call is tailoring. So both all these aspects are very critical to one when someone looks at that, what is how precisely we are detecting or peak of interest, and that greatly influences on your prediction of your analyte concentrations. So these factors need to be considered when someone looks at this the chromatography property, especially in terms of this peak and at the same time elution time. So depending on the nature of your separation techniques, depending on the nature of polarity, so elution compounds will elute at different times. So we need to look at that, whether your analyte of interest is eluting in such a fast, in such a way that it is actually going in the washout area. For example, the what we call in general here is void. So in that void period is nothing but when you are actually, suppose if you are for example 2 minutes or 5 minutes depending on your column and the detection limits, if you are running your runtime, if it is you are maintaining from 2 to 5 minutes, if anything between 0 to 1 minutes, if you are not getting any peak, probably there could be a choice that actually your peak of interest might be washed off during the initial phase, where it is going into noise, or because in plasma or serum or any other matrix, because of the even whatever you separate it out, it has its own inherent components which are not properly separated out, that still come and show the response. So normally those responses, because of the more polar in nature, they come very fast. Hence, the immediate either depending on the length of your runtime, the precisely the immediate 0 to 2 minutes, if it is a 10-minute runtime, I'm talking about that, considered to be a void time where your peak, your analyte of interest might go in the same time. So one needs to critically look at that, whether is a peak is missing, is it going in there, so that again we need to look at the separation. So such things need to be understood. At the same time, there are the properties like platelet count and capacity, which will determine your the peak shape and other aspects of that. So again, we need to be very vigilant how best I can reduce the time of elution. Again, if you are developing a method for example, if you are developing a method for 10 minutes runtime, or if you are developing a method for 20 minutes runtime, or you are developing a method for 1 hour time, it depends on the nature of the sample. Generally, 1 hour time and all people used to follow to very well separate, and maybe if they want to characterize any impurities or any particular metabolites or unknown peaks are coming into the your coming and colliding with your analyte of interest there. So widening of this elution time is very much important in a way that they can try to separate it out. But at the same time, because we in a particular pharmacokinetic study where you may need to generate or analyze to more than 200 samples per day, if you are putting a runtime very high, then it is the overall lead time of your analysis will go in a long time, such a way that your outcome will take long time, and any issues come out of this again going back to the either chemistry or design aspects of the formulation research, again it takes a longer time. Hence it is very important for us to reduce the runtime to the extent possible, but at the same time adequately and precisely we need to analyze the sample. So how best we can use either short columns, or maybe can we use instead of HPLC, can we use UPLC, where well separation but within the reduced runtime with the ultra performance can be characterized, so that the choice of these separation techniques can be tailored out in a way that you can reduce the runtime to the extent possible and analyze of your samples at the earliest possible. Okay, so these are the various phases one can look at, that how best we can reduce the time, so that at the same time reducing the time also will help not only the time, it also reduces your cost, because when you are keeping a length of your time like 10 minutes, 20 minutes, because you are injecting a lot of mobile phases you are running, which are continuously which are comprised of the organic solvents. So the wastage of the more number of the more volume of the organic solvent which you will be wasting. So if you reduce these time factors, definitely you can reduce the usage of the organic solvent to the extent possible. It is good for the environment as well as the cost. So with all these separation technique, one can look at that. And next phase is detection. What is the detector need to be understood? So depending on the nature of the sample again, or your concentrations, or the levels at which you will be estimating, one can look at that whether a simple spectrophotometer is good enough, which can be coupled to HPLC, or do we need a selectively and then faster estimation of your analyte of interest? Then can I use the mass spectrometer? Or depending on the volatility of the compound, can I use that HPLC instead of GCLC or GCMS systems? Or again, depending on the nature of the your analyte of interest, can we use like a TOF detector, time of flight detectors, instead of the regular MS by MS triple quadruple, or the ion trap detectors. So and why and again, there are the instances where one can analyze the samples and characterize the samples with respect to chemistry, and it can be coupled to LC, coupled to NMR. So that also is LCMS, and then it is coupled to NMR. So these are the various aspects, or various detectors can be used based on the need of your project, in such a way that you can detect precisely and accurately and rapidly. So at the same time, there are now a lot of widespread use of the ELISAs are coming in for the biological science and chemistry, because we are moving from the new chemical entities to the new biological entities, or biosimilars, or biotherapeutics, or non-biological complex drugs, where these ELISAs are also plays a role. So such that so a analyst should be aware of all these techniques in such a way that depending on the nature of the sample, nature of the requirement of the projects, one can look at that. Okay, what kind of the instrument need to be selected? What kind of detector need to be selected? So how we can, again, suppose if you are taking any UV detector, how we can rapidly detect our analyte of interest. So in early discovery, mostly people prefer to use the there are the monochromatic UV detectors, dual wavelength, and then multiple wavelength analyzers like diode array detectors. So one can look at that. Okay, can I use the PDE or the diode array detector coupled to HPLC, so that we can analyze parallelly, because rather than screening the compound for what's lambda max and then going back, so if at the screening stage, it would be very easy to use that as multiple wavelengths and then come out with the best suitable wavelength, lambda max, and where the response is high, and that can be, the data can be easily picked up. So one can look at that using of this PD or DAD detectors in the early phase, and during the development, again, they can look at that precisely for the single or dual wavelength detector, because you know that prior knowledge is there where the lambda max is there. So you can then you can look at that you are quantitatively how well you can precisely estimate, rather than getting because when you are using the multiple wavelength, then obviously other components can also interfere. Once if you selectively choose your wavelength, then the interfering peaks can be avoided to the extent possible, and so that you can precisely analyze your sample. So in the development, it will be mostly either singular dual wave length detectors will be used, and now in the current scenario, it will be mass detectors, where a triple quad will be regularly used. And one of the best classical instrument in the industry, which is used in the entire global analytical research, is the SciEx instrument. And out of that, API SciEx 4000, which is a triple quadruple mass detector, which is widely used unless until you have very sensitivity, like where you need to go down to the very low sensitive levels. Otherwise, it is a very good instrument, a rugged and robust instrument. So because it has three quadruples which are efficiently selectively trap the ion to the based on its ionization pattern, and then subject once it is trapped, and then those ions, selective ions, would be going to the next phase of the second quadruple, where it will be collided with a high energy procedures in such a way that it will be fragmented over there, such that once it is fragmented, but unfragmented and fragmented both will come out of that, and you can choose a most predominant and abundant fragment peak as a in addition to your unfragmented peak, so that it will be considered as, that's why we call it as MS by MS. So the your unfragmented by fragmented one, you can selectively choose your analyte of interest, and that will be, again, those will be trapped into the next quadruple, which is a Q3, what we call in the triple quadruple, and that will be taken into your converted into your signal, and you will get a very precise signals over there. So these triple quadruple mass detectors are really rugged, robust, and widely used in industry. At the same time, as I discussed that depending on the nature of the sample, if you want to go for metabolite characterization, if you want to go for any ionic separation, then depending on the nature of the sample and depending on, so TOF detectors are next choice of detector. Based on the time of flight, it will be, you can see in this current diagram where the just here, based on the time of flight, it will go into your analyte of interest will be separated out. The response will be, signal will be detected as per the flight of the time, so that you can very well separate it out the peak of interest from the unwanted peaks, or maybe multiple peaks. So these are all the various detection techniques, and one can look at that again efficiently, and how rapidly we can detect in such a way that, so these separation techniques, these cleaning the procedures, at the same time using the detector, these are all complementary each other in a way that the method, once it is developed very robustly, it can be, again, the method should be in a position that we should be tailor the method very easily in case if the any subsequent problems arises out of the method. So that's how one can look at that. These methods are being developed very efficiently. And the next part is, the major important part is, once the method is developed, how it is being validated, which is it's here, it's mostly of a regulatory science, in order to show that your method is reliable, robust, and reproducible. So these are the various parameters. One can go through that, all the parameters, like right from the selectivity, specificity, linearity, range, accuracy, precision, extraction, recovery, how well your sample is extracted well compared to the, from the matrix of interest to the neat sample, which is nothing but the aqueous standards. And other important factor is the matrix effect, where any molecule can, once it's subjected to the, especially when it is in the biological matrix, it can either enhance or suppress the ionic separation, means ionic separation in a way that it can impact on your the predicted response, such a way that, suppose for example, if you want to test that in a buffer, how the peaks are coming, how the response is coming, in presence of the matrix, how the response is coming, if it's completely different, there seems to be a matrix effect. To again look at that, the matrix effect, one needs to understand the what best sample preparation technique or sample separation techniques can be tailored out in a way that the ionic separation can be lowered, such that it will not impact on the subsequent prediction analysis. So the next comes is the dilution integrity. So because you will be dealing with that multiple levels of the drug in the plasma or any matrix, what you are estimating from once it is administered, your linearity range may be 1 nanogram to 1000 nanogram in a given system, but you might be getting a more than like a 10,000 nanogram per ml of the concentration. So how do you, so you cannot directly inject the sample, you need to dilute here. Once it is diluted, whether the matrix, whatever you are using, that the integrity of the matrix is getting disturbed because of the dilution in such a way that it will impact on your the concentration analysis. So one needs to look at that multiple times of dilution, in what dilution range it is integral, it is still integral in such a way that the estimated concentrations are actually calculable, and it can be equally compared in such a way that you need to establish a range of dilution, and accordingly your sample collection and dilutions can be suggested. And the other most important is stability of the samples, because you are right from the sample collection in the clinic or in the animal house, from the whether it's a preclinical or clinical area, right from the sample collection, how you are transferring the sample, how you are storing the sample, how you are again taking out the sample from the storage device, how you are during the processing of your sample, at what time, till what time it is placed on the bench you are working. Does it require ambient temperature? Does it require any ice? It can be maintained under ice, this procedure. So all these things, once someone wants to understand that, you need to understand stability of the molecule in various conditions. So these are the various stability of the sample, like benchtop stability, freeze thaw stability when it is being taken uh, from the deep freezer. So how many times you can freeze? Like normally it will be collected, like one sample can be collected, maybe 1 ml you will be collecting, but you may require only 200 microliter. But you can repeatedly, depending on the requirement, you may repeat the analysis as well. So how many times it can be thawed again? Because we cannot, once it is thawed, we cannot keep it back in such a way that it will lose its integrity of the matrix in such a way that it will impact on your rate of interest. So how many times it can be thawed? So that understanding of the stability during the at least 3 phases of the freeze-thaw cycles, normally we try to understand the stability for the molecule. Even if it is one time or two times, one needs to understand and then suggest um, suggest in the protocol upfront, saying that okay, this need to be applicable, and this need to be used in that particular fashion. So other thing is the stock solution, whether you are preparing stock solution freshly, or are you using the solution which are made of the yesterday or maybe of 1 month back. So because sometimes it is very difficult to daily prepare the stocks. Can I use that bulk stock? But we should know the stability of the stock. So one need to establish the stability, solution stability of the stocks, and at the same time, in a given matrix, what is the short term and long term stability? Because depending on the length of the your preclinical or clinical study, for example, if you are doing a preclinical study where these normally validated methods are applicable when it is being taken into the consideration of the safety studies, like either 14-day toxicity studies or 28 days toxicity or 3 month toxicity study. So during all these subacute toxicity studies, one need to understand that, okay, so how much time it is, the length of the study, and after that, how much time it will take for the shipment and then another respective analysis. So entire duration need to be well justified with understanding of the stability, so that if we come to know that okay, the stability is not great, then we can channelize or design our experiment, saying that okay, we cannot wait till the 28-day. We need to analyze whatever the sample is collected, and prior to 7 days or 14 days, depending on stability, we need to be analyzed. So that way, the protocol can be designed based on the subsequent understanding of this the stability of the either short term or long term. So this long term stability is must till the your entire length of the study. You need to need to establish and then define the way forward. So there are the various types of the validation people will follow across the globe. Generally, full kit, which is called full validation, and next comes is a partial validation and cross validations. The full validation consists of all these parameters what we have discussed when it's first time developed and then subsequently used for the similar kind of studies. And partial validation is if there is any tweak in this, for example, a slight change in the dynamic range of the linearity, a slight maybe we are changing a minor parameter which could or minorly impact. Those things we need to look for the partial validation. Again, it need to be minimally for selectivity, linearity, and accuracy precision need to be understood during this partial validation study. At the same time, when it is being developed in one species, for example, you have developed a method in mice plasma, and you want to develop again another method for the rat plasma. So based on the prior knowledge, partial validations can be used. At the same time, cross validations. Again, cross validations, whether with respect to multiple analytes are coming and then one needs to go back and look at that. Maybe do we need to, unless until it is not impacting on your peak of interest, whether cross validation can be done, or at the same time in presence of the other analyte, how it is behaving. So, and lab to lab transfers, all these things, these cross validations may be applicable. But in general practice, to the extent possible, one need to be ensured that, okay, if I can do the full validation and substantiate the method as a robust and rugged, then there won't be any questions from the agency in such a way that you are, whatever the levels you will be estimating out of this concentration analysis will be precisely can be taken for the further pharmacokinetic predictions. So finally is the sample analysis. It is again underlying the same principles of the system suitabilities, what quality controls you are using during the validation, as well as one needs to establish the quality controls to be make it acceptable for the run to be accepted. So generally quality controls will be designed in such a way that low quality LQC, MQC, and HQC, and those things need to once if you say that when we are using a one run to be passed, all the quality control samples are spreaded very adequately. At the same time, they should pass within 2/3 of the majority of the quality control samples should pass within the 15% of the criteria what we normally keep for the acceptance criteria of the samples. So except for the LLOQ where it can be 20%. And one can look at that how the slopes and intercepts are behaving. Is it different for the calibration standards? Is it behaving differently compared to validation? At the same time, how about the analyte to IS ratio it is behaving, and is there any abnormal dips or peaks are coming in the pharmacokinetic estimation? One can look at that. Is there any IS is playing a role in a way that because IS response should be consistent. So if the IS response is not consistent, then it could lead to the abnormal dips or peaks of the curve in such a way that it will impact on your result. So one can vigilantly look at that the IS response, and at the same time, are there any known metabolite peaks or unknown peaks are coming in the sample? If the method need to be relooked at that, if they are interfering with respect to your analyte of interest, because we don't know. In particular run it may show, and second run there might be a carryover effect. So one can critically look at that and then look at that. And the most important aspect during the sample analysis, incurred sample reanalysis, because we develop and validate the method while normally in taking a blank matrix and then spiking of our drug and then understanding the nature of the matrix as well as the sample. Whereas when you administer a drug to the any biological system, the in vivo samples which are having inherent this drug in a matrix, they might behave differently compared to the spiked extraneously spiked sample. So those are called incurred sample, and hence one needs to understand that by repeating this certain percentage, like 10 or 20% of the samples of the whole set of the samples what you are analyzing, and if the reproducible samples are coming, then the ISR is less than 50%. If they are behaving differently, one needs to look at that. Okay, again you need to tailor out the method. At the same time, the when you are repeating the analysis, what are the criteria to be considered? Is it peak response, or is any known or unknown factors? There are the certain norms and the acceptance criteria. One can look at that, and as per your standard operating procedure, you need to follow strictly with the defined procedures, and then do the repeat criteria, repeat analysis, and subsequently report the results. So the lastly is the reporting of the results, which will be converting into a pharmacokinetics takes this data and he will convert into the plasma concentration time, and subsequently he will estimate all the pharmacokinetic parameters. A bioanalytical scientist always limit himself while reporting to bioanalytical results only. He should not go beyond and try to report in such a way that these are the plasma concentration curves, these are the pharmacokinetic, because it will lead to the biases in such a way that again you will go back and look at this sample. So one need to be very critical when you are saying that my method is rugged, robust, no criteria is being failed everywhere. Without hesitation, he should report the results. He should not look at that how the curve look like, how the pharmacokinetic parameter look like, but that is a subsequent collaboration with the PK scientist you can try to understand, but not, it is not the primary responsibility of the bioanalytical scientist. So in the interest of the time, since it is taking much of the time, I'm trying to very less focus on the subsequent metabolite selectivity coverage analysis, which is nothing but, as we discussed, some of the metabolites of interest are unknown peaks, which you can try to collate and see that you can cluster them and then look at them. What are the possible by theory or by your known experiments, whether it's in silico, in vitro, or in vivo experiments, and then subsequently collate all the unknown peaks or the known peaks in such a way that you can look at whether pathway analysis could lead to a predicted metabolic pathway. So those things can be quantified using these our bioanalytical procedures, and subsequently it can be conveyed to the respective teams to understand further whether the metabolite of interest need to be quantified continuously. Is it active or not active? These things need to be subsequently understood by the respective collaborative scientist. Hence, early detection of these human metabolites is very much important, rather getting surprised at the time of clinical development. So there are the regulatory guidances across the globe are available. Hence, these are like, for example, all these guidances actually typically look for that what standards you are practicing in your lab, which is nothing but good laboratory practices. One need to adapt to the ICH guidelines, and they have to follow the good laboratory practices. So we have the NGLMA, which is a Good Laboratory Monitoring Authority of India. So, which is as per the ICH, can come and audit your lab and then certify your lab as a GLP. At the same time, what guidances one need to look at that from the perspective of the bioanalytical development or validations are. Over the decades, a lot of changes and a lot of debates happen across the industry in such a way that the current guidelines, the team across the bioanalytical team, global teams are following is the US FDA bioanalytical method validation guidelines of the May 2018, and EMA guidelines, which is 2012, and at the same time ANVISA for Brazil 2012, and ICH is coming out with a harmonized method, because a lot of couple of the changes you can still find from FDA to EMA and other factors. So ICH is trying to harmonize, and which has come a M10 draft guidance. It is still draft in place in 2019 February, and there is a new guidance from the Canada as well in 2008. So these are the guidances. One can refer to that and then follow adequately in compliance with these guidelines. They can validate their methods and then report the results. So there are the some more guidances in terms of metabolite monitoring also. So one can refer to the literature and then can find out these guidances, which are maybe helpful during the preclinical development as well as subsequent metabolite analysis, even for the clinical development as well. And nonetheless, now new guidances are also being published in terms of because we are moving from the NCE to NPE. A lot of peptide molecule, protein molecules are coming into play where we are getting analyze all these samples. So there are the new guidances for the immunogenicity. There are the methodologies have been well written in these guidances. How one can look at that the immunogenicity potential of the compound using those guidances can be referred to. So overall, I would like to conclude here that this bioanalytical support is very critical, not only for preclinical study, for clinical studies, and at the same time, it should be very rapid in order to support the overall development process. And once the method is developed, it should be sufficiently rugged and then such a way that it can lead to a longer time. At the same time, it should provide opportunity for either more tailoring of depending on the minor modification, ease of adaptability to suit to other needs of the method, such a way that it can support the pharmacokinetic and toxicokinetic studies very well during the entire characterization of the development program. At the same time, there is a need for the sound bioanalytical method by the scientists should be understood, but at the same time, scientists also should practice in a way that it's not only a regulatory science. One need to have a scientific rationale in such a way that their thought process should be practiced both in line with the regulatory as well as the scientific environment, such a way that it can be tailored out depending on the need, at the same time while following the regulatory science. So a bioanalytical scientist role is very, very important over here. Rather blindly following the methods, he should be visualize the method discrepancies, if at all. He should be able to troubleshoot the method, and then he can generate and support the entire development process in a very efficient manner. With all this, I would like to thank you all for patiently listening to my talk. At the same time, I would like to acknowledge my industry colleagues, my close friends who are working in different industries at various levels. And these are all, I can consider them as mentors in my bioanalytical area. And most of them are working at various different bioanalytical headship positions at different companies. Definitely, my special thanks to them. And thank you all.
Yeah, thank you, Kantikiran. It's a very nice informative session. The explanation we don't get in any of the textbooks. What you explained, it's really good. Uh, Now we will move on to. We have few questions in chat box. Can we move?
Yes, please.
Yeah, right. The first question is from Sugandha Mulgan. Can you please elaborate the actual procedure for determination of LOD and LOQ for analyte?
Yeah, sure. So as we discuss that, so one needs to differentiate the whatever the blank response. So a plasma blank or whatever the matrix you choose, what is the signal it is giving that? So the blank response, once if you can understand that, the LOD should be in such a way that at least the signal to noise ratio should be minimum 5 times, so that you can the signal quality of the lower limit of detection can be easily defined, so that the LOD can be defined in a quantified. It should be detectable. At the same time, when it comes to LOQ, at least 10 to 20 times, if you can make that the signal to noise ratio difference between the from your plasma blank, so that you can adequately and precisely quantify the levels of the analyte of interest without getting the interference of the blank matrices. So that's how the LOQ and LOD can be determined. At the same time, it depends on the sensitive, like what levels you are looking in the method. One needs to look at that. Okay, do I require to? Is it a requirement for my LOQ? Is whether I need to estimate at a 10 nanogram per ml or 10 picogram per ml. So depending on the requirement, one can again relook at that. What best separation techniques or what best like a sample preparation technique can be used in tailored way, or detect using the advanced detector. You can achieve the lower LOD and LOQ to the nearest extent possible based on your requirement.
Oh, good. Well, we have one more question from so participant. What guidelines to be followed for the validation of bioanalytical methods? So you answered it the last before that she posted that question. Yeah, you already answered that.
Yes, of course. One can refer to the guidelines, whatever is existing into that, right from the US FDA to across the ICH.
One more question we have. Can you explain the preparation of solutions for long-term stability used in toxicity studies? So maybe he's asking, yeah.
Yeah. Yes. So we need to consider one of the factors, especially during the toxicity studies, because you are using toxicity studies are generally used for the NCEs, okay, which are first time in nature, which is not very well established or approved drugs. So obviously, the limited amount of the knowledge uh, about that molecule is not adequate enough as that of the marketed products. So it is always better to use to the extent possible the fresh solutions, okay. At the same time, but there is a again being a NCE, there are the limitation with respect to quantities required and other things. So one can look at that. Okay, how precisely I can use that same solution. But so in general, it is if the material is available, easily synthesizable, it can be made every time the fresh solution. But if you want to use it for generally, it is like, for example, toxicity studies are meant for the short anywhere between the 14-day toxicity to 3 months toxicity in the prior to this phase 1 to phase 2 studies. So in a way that one needs to look at that, whether I can substantiate at least 7 days, or I can substantiate at least 14 days or 28 days, so that the dose formulation analysis, or at the same time, it can be even the stock solution can be used for the bioanalytical samples in such a way that it can be So we need to establish the first of all the stability, stock solution stability, and then decide the way forward and how much time below which we can use the same stock, whether at kept at room temperature or whether it kept at the 2 to 8 degrees. So those things need to be considered and then define the way forward.
Yeah, thank you. One more question we have. This question is it's a simple question, but always the teachers have question mark. Sir, what is the actual use of internal standard in bioanalytical method validation? I mean, bioanalytical method development and validation. So, can we develop without internal standard these bioanalytical methods, or is it mandatory that according to guidelines we need to use internal standard?
Yes, it's a good, definitely a good question.
Yeah, yeah.
Yes, okay. Yes, one can develop without internal standard when there is no adequate or suitable internal standard is available, and in the preliminary, very early discovery process, without having a proper, because once if you know that a series of compound core pharmacophore is established, one can look at that. Okay, what are the internal standards I can use or any similar kind. But without that, also people will try to analyze the sample. But the problem with that is, especially as I mentioned to you earlier during the discussion, that when you are analyzing the sample, because you are trying to estimate the drug which is not known in nature, and it varies from time to time with the different concentrations in a given sample. So at different concentration, it might impact on the again on the matrix in terms of the ionic suppression or ion enhancement, so that which can lead to again a wrong prediction of the estimation. So once if you identify a suitable internal standard, which and when you spike it across the samples, irrespective of what concentration of your analyte of interest, so across the samples at a defined uniform concentration, so that you can look at that. Is that when you want to look at that, is that predictable concentration is accurate or not? One can look at the IS response. If the IS response is good within that 15% and how it is behaving, then you can confidently say that yes, these are the predicted concentrations are behaving like this. Otherwise, it is always a challenge to comment on the unknown nature of it.
So, one more question we have. Uh, Column theory, can you brief it, sir? It's not clear. She's asking about column theory. Can you brief it? I mean, does she want to the type of stationary phases, like in terms of that, she may need.
Yeah. So I I'm basically a pharmacokinetician, and okay, so these are all like a bioanalytical techniques. I have developed the methods on my own for my work, as well as I have managed the systems. Definitely, as a manager and as a pharmacokinetician, I try to understand to the extent possible. But column chemistry is the area. Definitely, I will say that I do not have much knowledge in that particular aspect. I always use my bioanalytical scientists to give their feedback. And as I mentioned to you, that one needs to look at that based on the category, whether we are using a reverse phase chromatography or we are using a normal phase, depending on that, and then depending on the response, how we can troubleshoot that. At that time only, I could interfere along with our scientists, but I'm not an expert in that area. Sorry to say that.
Yeah, one more question. This is from the same participant who asked about LOD and LOQ. He just extended one more doubt. So after establishing the LOD and LOQ, uh, uh, in the range that has to be established to be linear, or they can fall outside this range. He's taking, you'll be setting the linearity range. So does it need to be within that range or out of?
Definitely, it need to be like within the linear range, because one need to, because we want to see unless until you come out with a regression line, and then you'll establish the regression line and then predict your concentration. It is very difficult. Yes, in general, for the synthetic compounds, it is established that it should be linear. But there are the other bioanalytical areas where you will be establishing the ligand binding interactions and other aspects where again the response could be like a sigmoidal shape and other things, where it can go up to the nonlinear range. But that is in majority of cases where in general practice to support an NCE or this formulation research, definitely it should be linear.
Yeah, thank you. One more question we have. Can you explain the bioanalytical method development of separation of enantiomers from racemic drug? So, is it possible or is it a tough job for developing a bioanalytical method for enantiomers?
It is possible because there are the specific columns are available to separate it out for the enantiomers. And by using again, mostly it goes with the normal phase, and then subsequently using those specific enantiomer separation columns, it is possible, and it need to be understood as well.
Yeah. Thank you very much for all your patient answering of questions. We have a lot of compliments for you, Kiran, about your session. So it's a very nice, informative session. All the participants are very much happy about your explanation part of sample preparation techniques and basic steps in bioanalytical method development. So nobody will accept that, like, no, even though you are a pharmacologist, right? So you explained in a better way than an analyst, right? So you had to agree with me in that aspect. So lastly, I asked one question from my side. Does anticoagulant, what we use in plasma preparation, does it affect the method?
Yes,
It-
ma'am.
Yeah.
Definitely. So probably you brought the right point because I didn't consider in my slides. So definitely that plays a major role. So it plays a major role in terms of the LCMS estimations because again it plays a major role in this ionic interactions in such a way that so one need to understand the one is anticoagulation and other one is hemolysis as well. Okay, these are the two aspects one need to understand, and accordingly we need to define our protocol for the collection of the samples.
Okay. Thank you, Kiran. Now I request my colleague Raghavendra Babu to propose vote of thanks to Dr. Kiran. Over to you, Raghavendra.
Raghavendra sir, please unmute.
Good afternoon to one and all. It is my honor to propose thanks note to Dr. Kantikiran Varanasi. I, N. Raghavendra Babu, on behalf of Organizing Committee of e-FDP program, express my sincere gratitude to Dr. Kiran for taking out some of his valuable time and enlightening us with his knowledge. Sir, your session was informative and helpful to all the faculty members to convey to their students. Thank you for throwing light upon the following topics like importance of bioanalysis in drug discovery and development. The way you explained the bioanalytical method development approach is very much useful for all the teachers and researchers. Sir, you explained the sample preparation techniques very clearly as it is a critical step in bioanalytical method development. So once again, we are very much thankful to you. Thank you, sir.
Thank you.
Yes, sir. Thank you very much, sir, for your enlightened talk on bioanalytical techniques. Thank you very much, sir. There is an important announcement regarding today's sessions and yesterday, tomorrow's sessions. Please visit G. Pulla College of Pharmacy, GPRCP Hyderabad YouTube channel for more updates. For assessment come feedback form, please visit www.gprcp.ac.in. Subscribe to GPRCP Hyderabad YouTube channel for more updates. And assessment come feedback link will be activated after 1 hour of this session, and it will be active for 24 hours only. And tomorrow, that is day 5, 12th July 2020, from 10:30 a.m. onwards, live stream will start on the topic role of in vitro in vivo correlation IV/IVC in bioequivalent studies and bioweaver by Dr. Vijay Kumar Nagabandi, Deputy Manager, New Hand Pharma Technologies Private Limited, Hyderabad. So once again, thanks to all the participants, organizing committees, and the Kranti Kumar sir, today's speaker Kranti Kumar sir, and the organizing committee of all the team members uh, of e-FDP Department of Pharmaceutics and Pharmaceutical Analysis, organized by G. Pulla College of Pharmacy and in association with Indian Pharmaceutical Association, Telangana State Branch. Thank you one and all.